COVID infections can produce severe inflammation and damage in pulmonary tissues. This inflammation can persist into the post-acute sequelae of COVID, known as PASC, or long COVID. In this YouTube, we review a single paper published in 2025, which describes novel uses of the calcitriol analog Paricalcitol, a vitamin D receptor agonist, to attenuate lung inflammation. We will relate this paper to a paper published in 2024, which shows that the CoV-2 spike protein fragment, the S1 fragment, can localize within pulmonary tissues as well as the skull, the CNS, and the kidneys. So the use of Paricalcitol, a vitamin D receptor agonist, as a means of attenuating inflammation transduced through the toll-like receptor 4 nuclear factor kappa B signaling axis, pertains to the use of Paricalcitol as an anti-inflammatory agent. So first, the title of this paper: Attenuating Amniotidrone-Induced Lung Toxicity by the Vitamin D Receptor Activator Paricalcitol in Rats, targeting toll-like receptor 4, nuclear factor kappa B, HIF-1 alpha, and TGF-beta SMAD signaling pathways. To begin with, let us define some nomenclature. Paricalcitol is a synthetic analog of the endogenous immune hormone calcitriol, the kidney metabolite of vitamin D. Paricalcitol is 125 vitamin D2. It is an abbreviation for 125 dihydroxy vitamin D2. The vitamin D2 molecule backbone is hydroxylated twice, once at the 1 carbon position, and the second additional hydroxylation is at the 25 carbon position. This molecule can be more conveniently abbreviated 125D2. This is the ligand which serves as an agonist for the vitamin D receptor, which is abbreviated VDR. In the center of the slide is a green information vector describing an anti-ferroptosis pathway which is transduced by the vitamin D receptor, VDR. An agonist such as 125 vitamin D3 calcitriol or 125 vitamin D2 Paricalcitol. Collectively, those can be represented by 125D, 125 dihydroxy vitamin D. Calcitriol or Paricalcitol can bind to the vitamin D receptor, activating it. The vitamin D receptor is a transcription factor. It can then upregulate the master anti-inflammatory antioxidant transcription factor, NRF2. NRF2 can upregulate the key antioxidant enzyme, glutathione peroxidase 4, GPX4. GPX4 chemically reduces reactive oxygen species as well as peroxidated lipids, which are key drivers of the programmed cell death pathway known as ferroptosis. We have discussed this pathway in previous YouTubes on this channel. We will link this signaling axis, this anti-oxidation, anti-ferroptosis action, to additional actions that are being described in this paper. In short, Paricalcitol has been found to have antioxidant, anti-inflammatory, and anti-fibrotic activities, and it is this last concept, anti-fibrotic activity, which bears on the treatment of COVID and long COVID patients to suppress, attenuate pulmonary fibrosis as a consequence of COVID-2 infections, particularly the prolonged presence of COVID toxins like spike protein fragments in pulmonary tissues. So first, let us describe what amiodarone is, its medical context, and its potential toxicities. Amiodarone is an antiarrhythmic medication used to treat and prevent a number of types of cardiac dysrhythmias. These include ventricular tachycardia, ventricular fibrillation, and wide complex tachycardia, atrial fibrillation, and paroxysmal supraventricular tachycardia. Evidence of benefit for cardiac arrest is poor. It can be given by mouth, intravenously, or interosteally. When used by mouth, it can take a few weeks for effects to begin. Common side effects include feeling tired, tremor, nausea, and constipation. As amiodarone can have serious side effects, it is mainly recommended for only significant ventricular arrhythmias. Serious side effects include lung toxicity, such as interstitial pneumonitis, liver problems, heart arrhythmias, vision problems, thyroid problems, and death. If taken during pregnancy or breastfeeding, it can cause problems in the fetus or the infant. It is an antiarrhythmic medication. It works partly by increasing the time before a heart cell can contract again. Now, reading from the abstract of this paper published in 2025, amiodarone, an antiarrhythmic drug, has been reported to precipitate lung injury by various mechanisms. Vitamin D receptor is extensively expressed in the lung, and disrupted vitamin D receptor signaling axis may underlie various lung disorders. Therefore, the current study intended to explore the beneficial impact of Paricalcitol, a vitamin D receptor activator, on amiodarone-provoked lung injury, and elucidate its possible involved molecular mechanisms. Male Windstar rats were intraperitoneally injected with Paricalcitol and orally administered amiodarone once daily for 4 weeks. Our findings revealed that Paricalcitol diminished BLAF leukocyte count and total protein, serum LDH activity, and pulmonary histopathological changes, and counteracted pulmonary oxidative stress. Moreover, Paricalcitol decreased pulmonary toll-like receptor 4, nuclear factor kappa B, tumor necrosis factor alpha, transforming growth factor beta, and phosphorylated small mothers against decapentaplegic 3, that's p-SMAD3 levels. All of this is in line with less lung fibrosis percentage. Interestingly, these results were accompanied by suppressed hypoxia-inducible factor 1 alpha, HIF-1 alpha, lung expression. Taken together, Paricalcitol protected against amiodarone-induced lung damage in rats through antioxidant, anti-inflammatory, and anti-fibrotic activities. Again, we are going to relate the findings of this paper published in 2025 to a broader set of literature which pertains to the pathogenesis of pulmonary tissues in COVID and long COVID patients. However, this same pathway and the anti-inflammation, antioxidant, anti-fibrotic activities of Paricalcitol also pertain to other tissues in IAC conditions, infection-associated chronic conditions. Before moving on, I'd like to draw your attention to the figure at the lower right of the slide. This is a paper published in 2024. This is a very important, iconic result for long COVID. The investigators took the S1 fragment of the CoV2 spike protein and conjugated a fluorescent molecule to it, so it's fluorescently labeled S1 fragment. They injected it intravenously into a rat. 30 minutes later, the rat was euthanized, then chemically cleared to make its tissues transparent. The entire rat was then imaged with a sophisticated imaging method called light sheet microscopy. And with this, you were able to see the distribution of the S1 fragment distributed within various tissues and organs of the body. It accumulates in the brain as well as the skull. It's also present in lungs, heart, and highly concentrated in the kidneys. So wherever the S1 fragment localizes, it can potentially bind to toll-like receptor 4 and initiate inflammatory activity. Unchecked, unimpeded, hyperinflammation can result in tissue damage and subsequent fibrosis. So in short, the study of pulmonary fibrosis in response to an antiarrhythmic drug, amniodrone, provides important molecular information about how paricalcitol could potentially protect various tissues, visceral tissues and nervous tissues, from the toxins that are produced by infectious agents, as well as autoimmune conditions.